Hey everybody, it's Miss Landry, and we are on to lesson 2 in our first unit, which is experimental design. So let's jump right into it. What are you going to be learning in this one? You're going to learn about the different types of experiments and the different types of science. You're going to learn about two very important variables, independent and dependent variables, which I know you guys have heard about in the past. You're going to learn about controls, positive and negative, and then we're just going to review. This should be pretty quick, but it's also very important. So make sure you have your notes ready to go. So let's start with the two different types. There's actually three, but we're going to call it two because you'll see why in just a sec. The first type of research is called basic research. Now, basic research is exactly what it sounds like, okay? It's the basis of our knowledge in science. It is not basic in terms of simple. It's just basic in that it's moving our knowledge forward, okay? And it's what helps the next type of science be able to do what they do. So examples would be, let's say that there's a new type of cancer we found and we want to know DNA's role in that cancer. So that's definitely basic science, or what we also sometimes refer to it as bench science, if you've ever heard that. What causes mutations? Think about, with all the cool stuff going on in space science, how does gravity affect plant growth, for example? Those are all bench science or basic science type of research. Then we have applied science. And this is something that is applied to the general population. So you can think of it as being clinical trials, right? So I'm testing a drug for treatment of cancer, and I need to know what side effects it's going to have, what dose is the best dose, all of those things are applied, right? Like it's actually being applied to a specific problem. Now, there is a third type. It's called translational research. And this is where you kind of take what we've learned and also the applied and bring them together. So it's kind of that intermediate step. For this class, I really just want you to know basic or bench and applied research. So let's talk about what a clinical trial is. So a clinical trial is actually pretty cool. Basically, what happens is after a research project has been completed in terms of does this actually work in animal models, then they're going to try it in human models. And so there's 4 phases. Now the first phase, phase 1, is where it's very small. Very few people are in this trial because they're still not sure about side effects. They're not sure about dosage, things like that. I will tell you that my mom was in one of these phase 1 trials for a cancer drug, and she was told right up front, like, this is not going to help you. This is more for the people that come after you. And so they were at least honest about that. Phase 2 is if phase 1 is successful, there's no real big dangers. Phase 2 gets larger, and so now we're starting to look at what dose is effective because we want the smallest dose with the highest amount of effectiveness. Phase 3 is now what we're doing is we're moving into an even larger population. We're going to actually start looking at different populations, so different countries, different genders, different ages, different health risks, things like that. And then finally, phase 4 is after it's been approved, then it goes into this long-term study. So now we're going to study it over a long period of time in many, many different populations. I love Madame Curie's quote that nothing in life is to be feared. It's only to be understood. Please know that when we are scared of something, it's generally because we don't know very much about it. And so knowledge and education is how you can combat fear. So now, as she says, now is the time to understand more so that we may fear less. Okay. Let's talk about the 3 variables I want you to know. First is the independent variable. So I want you to be able to not only choose a good one, but also identify it in a study. So let's talk about what the independent variable is. So I'm going to get a little pen here. So we're on this one right here, independent variable. So this is what you as a scientist are changing, Okay, so you think that this variable specifically is going to be the thing that causes an effect or has a correlation, and we'll talk about those in a minute. Now, in a good study, you should have only one independent variable, and that is because if you have more than one, you're not going to know which one it is that caused the effect. So, for example, if I have a really bad cold and I take like 4 different cold medicines, don't ever do that. I have no idea which one actually made me feel better. And so, really, you need only one variable. The other variable is a dependent variable. So, you can kind of think about this one as depending on the independent variable. So, you are going to be measuring this one, right? And you can have a ton of these. The more, the merrier. So, what effect do you think that your independent variable is going to have on the dependent variable? Finally, we have controls, positive and negative, and I think you'll learn a little bit more about those through examples. But basically, controls are going to be used for comparison to see if your independent variable actually did cause a change. So let's do an example. Let me erase the screen. So here, just take a second, pause the video, look at this, and I want you to tell me what is the control? I kind of gave you the answer. But take a look at this first plant here, okay? This first plant is not getting any fertilizer. The little green drops are fertilizer. So notice it gets no fertilizer, but it gets the same amount of water and the same amount of light. So that is considered a negative control, meaning it's not getting, aka negative, the independent variable change. Okay, so that's going to be your control. Now, the next question would be, what is the scientist changing, or what is your independent variable? So on your notes, go ahead and write down what you think it is. And then your dependent variable. So what is going to be affected by the change? What will you measure? Go ahead and pause and write your answer down. So for the independent variable, I was really looking for amount of fertilizer. If you notice, this one gets 2 drops, 4 drops, and 6 drops. So how much fertilizer it's getting. And then for the dependent variable, it could be anything. This picture doesn't really indicate, but you could look at height, you could look at number of leaves, you could look at mass, like how much mass the plant has. You could look at the health of the leaves by color, things like that. Hopefully that made sense. So we have control, this first one, independent, the amount of fertilizer, and dependent is what we're measuring. So let's try one more, and you're going to do this on your sheet. So pause the video, and I want you to answer, what do you think the research question is and the hypothesis? What's the independent, the dependent, and do they have a control? So pause the video. I'm not going to go over it on the video. We're going to go over it in class. So just in summary, we went over the different types of research. We talked about the 4 phases of clinical trials. Excuse me. We talked about how positive and negative controls are used to compare. We talked about how the independent variable is the one that you, as the scientist, will be changing. We talked about dependent variables and how you're going to measure them. And then we did a few examples. So make sure you have your notes for class, and I will see you guys soon. Have a wonderful evening.